Hello, my name is Cheyenne, and my presentation is going to be on photosynthesis. So what is photosynthesis? Photosynthesis is the process used by organisms to convert light energy into chemical energy that can later be released to fuel organisms' activities. This chemical energy is stored in carbohydrate molecules such as sugars, which are synthesized from carbon dioxide and water. Photosynthesis is found in plants, bacteria, and some protists, and works similarly throughout all these organisms. The equation for photosynthesis is as follows. 6 carbon dioxide molecules plus 6 water molecules in the presence of light create a molecule of glucose and 6 oxygen molecules. However, this equation is only a summarization of the multiple processes that actually take place during photosynthesis, with only some of the components and products of photosynthesis. But before we start with how photosynthesis works, let's talk about how it began. Photosynthesis first showed up in prokaryotic cells called cyanobacteria 3.5 to 2.5 billion years ago. These cells helped to release oxygen into the atmosphere after evolving simple organelle-like structures that helped harvest light when they floated at the surface of oceans. The release of oxygen into the atmosphere led to mass extinction of many anaerobic cells that were negatively affected by the oxygen. This is known as the Great Oxygenation Event, and the reaction of oxygen and the methane already present in the air led to a thinner atmosphere that caused one of the longest ice ages in Earth's history. After oxygen concentration stabilized, however, eukaryotic photosynthetic organisms started to appear. It's believed that a prehistoric microbe swallowed a cyanobacteria whole and used it as a source and factory for energy in a process called endosymbiosis. Today, the cyanobacteria is a chloroplast that is present in plant cells. This helped to diversify life on Earth, since the oxygen atmosphere allowed for the emergence of new and more complex organisms. Photosynthetic organisms were also special in that they were able to produce their own energy with almost unlimited supplies of raw materials, unlike anaerobic cells. Oxygen also allowed for the emergence of animal life on Earth because of how it helped in the evolution of cellular respiration. So we start off with C3 photosynthesis, the most common type of photosynthesis. Photosynthesis could be divided into two steps: the light reactions and the dark reactions, or the Kelvin cycle. The light reactions are reactions that occur in photosynthesis in which energy in the form of light is absorbed and converted to chemical energy in the form of ATP. The dark reactions are the reactions that involve fixation of carbon dioxide and the creation of carbohydrates such as glucose and the dissociation of water, using chemical energy stored in ATP to help create glucose. Chloroplasts and pigments play an important role in photosynthesis, especially in plants. The light-dependent reactions take place in the thylakoid membrane. The thylakoid contains pigments, usually chlorophyll, that absorb certain wavelengths. Chlorophyll A and chlorophyll B are present in the thylakoid, and both of them reflect green wavelengths, and is the reason why most plants are green. Chlorophyll B is known as an accessory pigment and works alongside with chlorophyll A, allowing for more wavelengths to be absorbed that chlorophyll A could not absorb. The light cycle starts off with a photon of light hitting a photosystem. A photosystem is what receives the photon of light to help excite an electron in a chlorophyll molecule to help in the transport chain for a light cycle. So the chlorophyll molecules, when hit with photons, help excite an electron, and when it returns to its ground state, it releases energy in the form of heat and light. This is important because this is what helps start the light cycle. The photosystem helps by using these excited electrons, and when they're excited, it has a chance of taking the electron into the electron acceptor and possibly through the electron chain through the light cycle. So the reaction starts with light hitting photosystem 2, and the reason it's not called photosystem 1 is because this photosystem was discovered first and we called it photosystem 1. So photosystem 2 helps in the photolysis of water or splitting of a water molecule to help replenish electrons for a transport chain. The electron is excited and oxygen is a byproduct, which is the oxygen animals and humans breathe, and 2 hydrogens are released into the thionyl lumen. The excited electrons are then transferred through electron carriers that become increasingly electronegative as it continues. As these electrons, however, pass through the carriers, hydrogen ions from the stroma are brought into the lumen. This helps to make the thionyl lumen more acidic because of the greater amount of hydrogen ions, while the stroma becomes increasingly basic as hydrogen ions are brought into the lumen. The excited electrons enter photosystem 1, and another photon of light helps excite the electron once more. NADP+, an important molecule, is an electron acceptor and accepts hydrogen to form NADPH in the presence of an enzyme called NADP reductase. NADPH will be used in the Calvin cycle later. The positively charged thionyl lumen contains many hydrogen ions, building a gradient, and the ions are pumped through a protein channel named ATP synthase across its concentration gradient. This gradient allows for the movement of hydrogen ions into the stroma. When the hydrogen ions are pumped through the ATP synthase channel from the lumen to the stroma, the energy in the process from the hydrogen ion allows the inorganic phosphate and ADP in the stroma to become ATP in the process of photophosphorylation. The ATP and NADPH will now be used in the stroma for the Calvin cycle. So the Calvin cycle or dark cycle consists of 3 parts: fixation, reduction, and regeneration. In carbon fixation, RUBP, a 5-carbon molecule present in the organism, will be attached to a carbon dioxide, and in the presence of an enzyme called rubisco, the most abundant enzyme on Earth, it will help to break it down into 2 3-carbon molecules called PGA. In the reduction phase, ATP and NADPH provide a phosphate group and a hydrogen ion, respectively, to make 6 G3Ps, which are 3-carbon molecules. And of these 6 G3Ps, 5 will be used in the Calvin cycle, and 1 will be used for other purposes. The 5 G3Ps that are used in the regeneration cycle help to create RUBP, the original 5-carbon molecule, and this helps to restart the Calvin cycle. The ADP and NADP plus made in reduction and regeneration can now be reused again for a light cycle, and the 1 G3P that was left over can be used to create sugars such as glucose, sucrose, or maltose. So the light reaction starts off with water, ADP, and NADP plus, and outputs oxygen, ATP, and NADPH at the thionyl, specifically in the thionyl membrane. The Calvin cycle needs carbon dioxide, ATP and NADPH created from the light reactions, and output G3P, ADP, and NADP+. G3P helps in creating glucose and restarting the Calvin cycle, and the ADP and NADP+ are recycled again for light reactions, and this takes place at the stroma. As you can see in the figure below, light and 12H2O enter the light-dependent reactions, 6 oxygen come out, the ATP and NADPH goes into the Calvin cycle, it helps to create carbohydrates as well as water, and the ATP and NADP plus that are left over go back to the light-dependent reactions to help restart the process of photosynthesis. We talked about C3 plants, but there are two other linear photosynthetic pathways. C3 plants have no special features to combat photorespiration, but C4 plants help to minimize photorespiration by separating the light reaction and the Calvin cycle, performing these steps in different types of cells. CAM plants, or known as crustacean acid metabolism plants, minimize photorespiration and save water by separating both steps by time between night and day. If you don't know what photorespiration is, it's when plants in plant metabolism, with the help of rubisco, oxygenate RUBP instead of using carbon dioxide. The products of photorespiration does nothing for the plant and actually wastes energy, with the net effect being losing 3 carbon dioxides. The reason photorespiration can happen is because on hot days stomata are closed, which stops water from leaving, but it also prevents carbon dioxide from entering, and so the plant is forced to use oxygen in the Calvin cycle. However, C4 and CAM plants are adapted to photorespiration. C4 photosynthesis is where the light-dependent reactions and the Calvin cycle are separated, with light-dependent reactions occurring in the mesophyll cells, which are spongy tissues in the middle of the leaf, and the Calvin cycle occurring in cells called bundle sheath cells. Carbon dioxide enters the mesophyll cell, and through a series of steps, malate is broken down into carbon dioxide and goes through the Calvin cycle and produces the necessary sugars for sustaining life. Malate is an organic acid which will be broken down for CO2. But the thing is, C4 plants use more ATP. However, it does decrease the effects of photorespiration. Next is CAM photosynthesis. They work almost exactly like C4 plants, but instead they take place in one mesophyll cell and divide the light and dark reactions temporally, which means by day and night, while C4 reactions are separated physically. Carbon dioxide is stored as an organic acid at night in malate and undergoes the Calvin cycle during the day after being broken down. CAM plants are especially effective in conserving water because stomata are open at night and closed in the morning. However, like C4 plants, more ATP is necessary, but plants like this are only usually found in dry, arid climates, and since they help to reduce photorespiration, it's worth the extra ATP. The 3 photosynthetic pathways I've talked about are all linear photosynthetic pathways, but there is another type of photosynthetic pathway called a non-cyclic phosphorylation pathway. So non-cyclic cycles are usually seen in prokaryotic autotrophs and sometimes in plants. So in this process, electrons repeatedly cycle through photosystem 1 without photosystem 2 being used. The cyclic pathway does not make NADPH since the electrons don't pass through NADP plus reductase, and since there's no Calvin cycle, since there's no photosystem 2, no oxygen is made. The reason this process could be used in plants is because plants might need lots of ATP, and this is a quick and easy method of creating lots of ATP. Or if the plant doesn't have enough NADP+, it could conserve it by carrying out non-cyclic phosphorylation instead of linear phosphorylation. So this is the end of the presentation. Thank you for listening.